Okay. So I hope you have enjoyed the conference so far. Now we're gonna move to the last keynote block of USS 2023, which would be from machines to brains. With that, our first speaker, the keynote speaker, Dr. Shahab Bakhtiari is here. Dr. Shahab is an assistant professor at University of Montreal. He received his undergraduate and graduate degrees in electrical engineering from University of Tehran. He then went on to earn a PhD in neuroscience from McGill University. After completing his PhD, he was a postdoctoral fellow at Mila, where he focused on research at the intersection of neuroscience and AI. He studies visual perception and learning in both biological brains and artificial neural networks. He uses deep learning as a computational framework to model learning and perception in the brain, and leverages our understanding of the nervous system to create more biologically inspired AI. The floor is yours.
Okay, so I'll just set up the talk. Okay. So, hi everyone. Thank you very much for the invitation. I'm very glad to be here with you today. As one of my colleagues said, the best thing that can happen to scientists is to be invited by students. So I'm very happy to be here. So today I'm going to talk about self-supervised learning and in AI. And specifically, I will talk about how this framework of self-supervised learning can be used to model and understand visual development, and ideally how we can take inspirations from visual development to develop better AI. So I will, so this is basically a pathway that we'll try to draw for you during the talk today. And basically I will describe some of the previous work that we have done and other people have done related to this topic. Okay, so it is very difficult, if not impossible, to ignore the recent advances in AI. So we have text-to-image models that, with a very short description of a text, these models can generate images that are quite accurately following the text-based description. You can see that how interestingly it can put together components that it takes from the text, put them together in the image in a way that makes sense to us humans. So these are, this is an example of a text-to-image model, AI model. We know that recent work has shown that you can actually take these so-called large language models and embed them in a robot, which then can follow instructions to do stuff for us, right? And we have chatbots, of course, our good friend ChatGPT, that shows behavior that from our perspective, we can consider it as intelligent behavior, right? But under the hood of all these developments are a few components that when you want to design such model, you have to follow those components, you have to design those components in order to give you the intelligent behavior that you desire, right? So what are those components? We have the architecture, right? So there are different types of neural network architectures. We have convolutional neural networks, multi-layer perceptrons, and most recently, we have transformers that basically underlie the recent advances in the last couple of years, right? So we put together this architecture of neural networks, and then we have the objective function, right? The objective function is either a loss function, a reward function, or something else that you maximize, minimize, in order to push the neural network to learn and show the intelligent behavior that you want. Of course, there's the learning rule, which is for 99.9% of the recent work in AI, it has been backpropagation using gradient descent. But, potentially, theoretically, you could change that learning rule as well. And of course, at the end, you have the data set. The data set that you basically use to train these neural networks for the desired behavior that you're hoping for. So when you put together all these components, you will have a deep neural network that ideally shows the behavior that you desire and you want the model to show. And this goes back to 70s and 80s, to the movement in cognitive science and AI that was, back then it was mostly known as connectionism and parallel distributed processing, and was well exemplified in these 2 volumes of book written in 80s. So one of them, the blue one, was describing all the advances back then in developing these models, training them, all the algorithm that had been developed by then. And the red one was interestingly describing how these neural networks could be used for cognitive science and brain sciences, right? So this is something that really goes back to 70s and 80s until the AI winter happened, right? And it happened in 90s when these models couldn't really meet the expectation of people, right? So this whole expectation has been built that these models can do crazy stuff. And then something happened in 80s. They kind of saturated on their performance. And for about 20, maybe 30 years, they were not the focus of attention for the AI and machine learning community. But then the AlexNet moment happened in 2012. A group of scientists from Jeff Hinton's lab in University of Toronto, led by Alex Krzyzewski. This is not 2019, this is 2012, sorry. So they basically took a convolutional neural network, they scaled it up way larger than the previous models, and for the first time trained these networks on graphical processing units and GPUs, something that we use very commonly today. But that was the first time that they used a GPU, they used its compute power to train a large, based on the standards of that time, a large convolutional neural network. They also leveraged the ImageNet database, which was, which it is still a huge data set, millions of images with their classification labels. They leveraged this compute and data in order to, by a very large margin, improve the performance of these models compared to the previous models. So this was basically the moment that ignited a revival of deep learning and artificial neural networks. And everyone started, again, paying attention to neural networks until today. Now you can, because of that, we see all the obsession and craze about deep learning because it kind of works. And since then, people have been playing with the architecture, with the data sets and training paradigms, and the models have been improving until today. They can reach almost 100% top 5 accuracy on the ImageNet dataset. So these models were quite successful, but at the same time, around 2013-14, people have started comparing these models, such as AlexNet, with the visual system of humans, right? Visual system of animals in general, primates in particular. And so they were focused on this part of you see my cursor? Yes. Okay. So they were focused on this part of the visual system known as the ventral pathway, which we knew for a long time that it was in charge of object categorization, object recognition, scene understanding, face processing, and so on. And people have been studying this pathway of the visual system for a long time. And there has been efforts for modeling this pathway to understand it and also to mimic its capabilities. But in 2014, people from different places started comparing models like AlexNet that are only trained to do object categorization, and they compared them with this ventral pathway of the visual system to see how well they can model this pathway. And the results were quite amazing. So this is how they did the comparison. So this is type of comparison that you will see throughout this talk multiple times. So you could record from the neurons in the ventral pathway of monkeys or other animals while they're looking at different natural images. You can record the responses of the neurons. So these black traces that you see underneath the red trace here is the recording from all these neurons. And the problem was to find a model that could actually predict this pattern of responses. And the red trace here, for example, you're seeing that this red trace is the responses of a model like AlexNet to the same images. So the model was quite capable of reproducing and predicting the responses of the neurons to this set of natural images. So the model was able to to replicate some functional properties of the visual system in monkeys. And, you know, again, that was in 2014. And now we have these benchmarks like BrainScore, in which you can submit your model, your desired model, any type of neural network that you trained for different purposes, different objective functions. You can submit that model to this benchmark, and that can actually rank that model with reference to the data from the ventral pathway of monkeys, right? And when you really dig deep into this benchmark, you can see that most changes since AlexNet has been in the architecture of the models. The models have become more complicated in architecture, and also the size of the models. Now, with the standards of today, AlexNet is considered a tiny model, right? So this has been the recent progress in comparing artificial neural networks with visual system. And there has been some formula behind all these models, like 90%, majority of the models. There is a convolutional neural network, there is supervised learning, and there's object categorization as the objective of training, right? So one important component of this whole formula is supervised learning, right? And as you know, supervised learning only needs pairs of inputs and outputs, right? If you want to train a classifier of cat-dog classification, you need images of cats, images of dogs, and a tag or a label that says, in this image we have a cat, in this image we have a dog, right? So we need label data for this purpose. And label data is expensive because, you know, all these data sets are labeled by humans, and it requires hours and hours of human work. So it's expensive. And most importantly, which is kind of the focus of today's talk, is that this is not how animals learn to see the world, right? So the way that kids learn to see the world is by acting in the world and learning from the stream of visual input in a self-supervised or unsupervised fashion, okay? So it's not like every kid is accompanied by an adult in every moment of their life, which, you know, the adult gives a tag for every visual experience that the kid has. This is not how kids develop visual capabilities. So there is a self-supervised way of learning visual capabilities. And ideally, we want to mimic that visual developmental process, because we can have a good model of visual development, which then will become useful for developmental problems. And also, we can ideally take inspirations from that visual developmental process to develop better AI, right? So this is why we want to move toward developing self-supervised learning models of visual development. So from here, I will start talking about how we need to do that, and what have been done before, and what are the next steps that need to be done in order to get there, right? So I start with some characteristics and constraints of animals' visual development. There are specific features that development has, and any model of development has to follow that, right? So I talk about 4 things, maybe 3, because I might run out of time, but I will talk about 3 or 4. And but this list actually is quite long. If you go and dig into the literature of kids development or animals development, you can actually extract many of these items to be added here. But let's talk about these. First of all, there's no explicit supervision, as I said, for visual development, right? Kids are not getting feedback for every visual experience that they have. There's a specialization. I'll talk about that. And there's neuroanatomical determinism and temporal autocorrelation of input stream. We will talk about that one by one. So how could the visual system develop without explicit supervisory signal? This is the main problem of self-supervised learning in AI, right? So you want to be able, the model should be able to learn representations directly from raw data without any label or tag that are then useful for some tasks or behaviors, right? The raw data can be anything. It can be audio signals, it can be images, videos, text, whatever, right? So as an example, we need, this model should be able to learn representations from raw images, okay? A huge data set of images, let's say the ImageNet database, but without the labels, just the images. It should be able to learn representations from raw images that then can be used for classifying objects, detecting objects, controlling them, etc. right? So during the learning process, the model is not seeing the labels, it's not seeing any kind of information for downstream behavior. It is trained in self-supervised learning fashion, but then it needs to be useful for some tasks like classification. Like any other deep learning approach, you need to design these components for self-supervised learning, right? So you need to specify the architecture, right? You need to specify the objective. And objective here is quite important because here we don't deal with a supervised learning problem, right? There is no input-output data pairs. Here we only have input. So it's important that what is the objective of training this neural network? And then of course the learning rule. So let's start with examples. And this is, this is a problem that people have been talking about for decades, right? And one of the earliest approaches to self-supervised learning or unsupervised learning, you can call it whatever you want, was autoencoders, right? So the autoencoders are getting raw data as input, and they're reproducing the input at the output. So you have an encoder, let's say a convolutional neural network again, that gets images as the input. It compresses them to some latent representation and then uses a decoder, another convolutional neural network, to reconstruct the input from that Z latent variable. When the model is trained, then you can take that latent representation of the data and use it for downstream behavior, for object categorization, for detecting motion, whatever task that you are interested in. And the objective here is reconstruction error. So this was one of the ideas that people came up with to train a model in a non-supervised or self-supervised fashion. But it is a good model. It has specifically, more recently, people have developed variations of this model that are very successful in learning good representations and also reconstructing the input quite well and generating samples. But one of the problems with this approach is that it has an encoder and it has a decoder, and both are quite large. So you can imagine that training this model can be quite a headache. So people came up with more with some interesting approaches more recently. One of them that I focus on now is now referred to as joint embedding parallel architectures. So here you don't have any reconstruction of input. Instead, you have a neural network, a convolutional neural network shown by CNN here. It gets an image as the input. It compresses it to some latent representation. Let's call it Z1, sorry. And there's another one. Now, here's an important point. This one here is not actually a different neural network. In 99% of cases, it's the same network as here, right? So you're not having two neural networks trained at the same time. This is almost the same neural network. So this second one is receiving a transformed version of the same image. And then the network forms a latent representation Z2, and then the network is trained to maximize the similarity between Z1 and Z2. So this is the objective function, maximizing the similarity between an image and a transformed version of the image. So here's an example. This neural network takes an image of a bird as input. The other network, which is basically the same network, takes a transformed version, here a rotated version of the same input. Same image as input, and the network is trained to maximize the similarity between the bird and the rotated bird. So what the network learns here is to basically ignore any type of information in the image that is affected by rotation. So the network is trained to be invariant to rotation. In other words, what the network will learn from this process is anything in the image that is not affected by rotation. One example is the identity of the object in the image. First image, second image, there's a bird there. So through this trick, we force the network to learn something about objects, ignoring all the variations that might happen from one object to another, from different instances of an object. Another example, which is from the same nature, same idea, here, the network, the CNN, is getting different frames of a video, forming latent representations of these videos, these frames, and trying to predict what happens next in the video. Okay, so here there's no transformation. The only objective is to maximize the similarity between the prediction of next frame and the actual next frame. Again, here you don't have any reconstruction of input. It is the same basically everything is happening in the latent space, which is one of the advantages of this model. Also, one source of problem, I'll not get into that, but these models might learn trivial stuff, and there have been tricks developed to avoid learning trivial representations. And people have shown that these modern self-supervised learning approaches actually work. Now, the state of the art can reach close to 80% top one accuracy, which is not that far from supervised learning on ImageNet. So in a nutshell, what I want to show here is that self-supervised learning in its current version and its current development, they work. They learn stuff that are useful for visual tasks. So are these models also good models of the visual system? So for supervised learning, like AlexNet trained on ImageNet in a supervised fashion, we showed that it was a good model of the visual system. But are these self-supervised learning models also good? So in the last 2-3 years, people have compared these models with different species, with humans, monkeys, and mice. And basically, all the results point to the direction that these models are also good models of the visual system. So there has been comparisons with the ventral pathway of human visual system. And as shown in blue here, it kind of actually, it's almost at the same level of performance with the supervised learning model. In another work in monkeys, if you look at this figure, for example, this plot here, the red bars are showing self-supervised learning models. The black one is supervised learning model, and it is at the same performance as a supervised model. And more interestingly, when people compared self-supervised learning with mice, they actually observed that self-supervised learning is way better than supervised learning for modeling mouse visual cortex. So there's something interesting and kind of unknown yet, that self-supervised learning is a better protocol for modeling mouse visual system than supervised learning. So what I showed so far is basically giving us some evidence that without any explicit supervision, you can learn good visual representation. We can develop good visual systems with useful representations. Now, specialization. So what we know about brains in general is that different parts of the brain, different anatomical locations in the brain are known to be kind of specialized for different purposes. And this is also true for the visual system. So how can visual development generate specialized pathways? Okay, how can a network that is trained with self-supervised learning generate parts within the network that are specialized for different purposes? So in the visual system, a mammalian visual system, we know that there are two pathways: the ventral pathway that I already mentioned in the previous works, and there's also the dorsal pathway, right? And there are specialists for different things. The ventral pathway is more about object recognition, categorization, scene understanding, dealing with the static contents of images. And the dorsal pathway is more in charge of localizing objects, tracking moving stuff, guiding behavior, guiding actions, using visual input, and so on. So they have different specializations. Now, if you want a neural network, a self-supervised learning model, it should also have the same specialization at the end of the training, right? This is the ideal situation. So we tested one version of self-supervised learning models to see if they can learn specialized pathways. So we took this predictive self-supervised learning model, so-called contrastive predictive coding is its name basically. But the idea is what I described previously. So the model is just trained to predict what happens next in a video. Okay, everything happening in the latent space, all the parameters of the model are trained end to end. So the model has different components, but this convolutional component here is the part that we use as the model of the visual system. So we train every part of the model, and then we take this convolutional neural network as the model of the visual system, compare it with different data sets from the visual system. So what we did in this work was that we took an architecture, a convolutional neural network that had two parallel pathways. And these pathways were segregated and identical. There was no difference between them. And we basically put this architecture here in the convolutional part and trained the model for doing prediction. And what we observed was that, and also we compared the network with mouse visual system, which we know like other mammalian visual system has this kind of ventral dorsal specialization. We compared our model with this visual system. And we observed that the two pathways do learn different representations. So they become specialized, and also they learn different types of specializations. So one of them becomes more similar to the ventral areas of mouse visual system. The other one becomes more similar to dorsal areas of mouse visual system. So long story short, the model, what our study is showing is some preliminary evidence that self-supervised learning is actually able to develop a specialization in a neural network if we use the right objective function and with the right architecture of the network. So specialization is also something that these models can develop. Now let's talk about neuroanatomical determinism. So this is a term that I'm using. If you look it up, probably you'll only find references to my other talks. No one has ever talked about neuroanatomical determinism, but what it means is that, if you take many animals, instances of the same species, and if you go through the ethical process and get the permission to open their brains and look at the specialization of this dorsal part of the brain, the visual system, you will always see the same functional specialization. If you go and record from another part of the brain across individuals with a good accuracy, you will always see the same specialization. So this is why I'm calling this neuroanatomical determinism. There's no randomness in the neuroanatomical location of a specialization. Okay, so but this is not the case in our model, in the two pathway model that I just described. Here, the pathways are identical. They go through the processing, and at every initialization of training, one of the pathway takes one specialization and the other pathway takes the other specialization. And it can change, it can switch from one training to another training. This is not like how humans develop, right? So we need to find a way to fix this sort of randomness in neuroanatomical location of a specialization. So this is basically, this data is just showing what I described. If I train a model, train a model, and measure the dorsal score of one of the pathways and calculate the difference between them, you can see that across initialization, the data is all over the place. In some of the initialization, path 1 becomes a dorsal pathway. In another initialization, path 2 becomes the dorsal pathway. So it is just random from one trial to another trial. Given that the two pathways are similar, there is some sort of symmetry breaking that happens throughout training, which is not quite understood. So we said, okay, if you want to fix the location of a specialization throughout self-supervised training, what can we do? We tried different things. So I'm not going to describe all the different approaches, but I'm going to talk very briefly about one of the approaches that is quite promising. So we said, okay, at initiation, one of the pathways should be pushed slightly toward dorsal-like specialization. If there is one of the pathways that I want it to become the dorsal-like pathway, I have to push it slightly at the initialization to become the dorsal-like pathway. The way we did it was that we said, okay, let's first take one of the pathways, pre-train on self-motion estimation. So self-motion estimation, just to describe what it means, when you're walking around, okay, you are getting a stream of visual input, right? And you can infer your direction of motion, your velocity, and so on, based on this stream of optic flow, this visual input, right? We showed in a previous study that networks that are trained on self-motion estimation are good models of the dorsal pathway, okay? So we said, okay, we're one epoch training one of the pathways on self-motion estimation so that we can trigger it a bit and push it toward dorsal-like specialization. Then we put the two pathways together and train it with self-supervised learning. So let's say we do it with path 1. We slightly push it toward a specialization for dorsal type of specializations, then put them together and train the two pathways together with self-supervised learning. And what we see is that in the majority of initializations, the pathway that was initialized with some dorsal specialization became the dorsal-like pathway. Let's do it with the other pathway. now you can see that the same happens, right? The pathway that is initialized to have some dorsal properties, that pathway after training with self-supervised learning with good accuracy becomes the dorsal like pathway. So the way we think about it is that this initialization, you can think about it as an algorithmic model of genetically induced asymmetries in brain circuits. So it's a mouthful, but it basically what it means that genetically encoded, there are some asymmetries in different parts of the brain that then kind of initializes those circuits to learn or develop specific functional specializations. And this is where the role of evolution can come in. And this is a path that we are following at the moment. Okay, so there are some hopes for neuroanatomical determinism as well. And the last one, which I'll not go over, go through it in detail, but long story short, you know, kids, when they're moving around, there's a lot of autocorrelation in their visual experience, right? Every moment of visual experience is quite similar to the next moment, right? So this is the autocorrelation in the stream of visual inputs. We know that artificial neural networks are actually quite bad from learning for learning from autocorrelated data. They have to deal with some, you know, all sorts of problems like catastrophic forgetting, not learning any useful representations from autocorrelated data. So if we want to develop a model of visual development, we have to make sure that that model is also able to deal with autocorrelated data. And this is a branch of work in the community now. So what I'm proposing is that we need to move toward a self-supervised learning model of visual development. So in order to do that, we formulate the constraints and requirements of visual development. This is what I did so far in that 3-4 things that I listed, right? There are things that we know about visual system and visual development that we can formulate them, we can describe them. And then we need to add those constraints to self-supervised learning models of visual development, okay? We have to make sure that our models of visual development can abide by those constraints. And this will narrow down the space of possible self-supervised learning models of visual development. When we have like 10 models that all can satisfy this criteria, then we can go ahead and compare them and see which one does better in modeling visual development. So what are the criteria for a good model? First of all, the model should learn useful representations of the world in the sense that it should be able to do stuff in the world. It should be able to control things. It should be able to do visual tasks, right? It should be useful. The model should learn representations similar to those of animals' visual system. I talked about that. And last one, which I think is the toughest criteria, is that the learning trajectory should align well with the developmental trajectory. So what does that mean? So far, we have been comparing snapshots of brains and neural networks. We took an adult brain, we took a well-trained neural network, compared them in terms of their representations. And we said, okay, this is a good model of the visual system, this is a bad model of the visual system, right? What we actually need to do now is that we need to take a look at visual representation through development of animals and humans, and look at the neural networks during training. Okay, so this would be the learning process of a neural network. This would be the trajectory of development of a visual system in an animal. And what we need to do is that basically we can form a trajectory of this learning process in humans and neural networks, and we can come up with the best configuration of a neural network and self-supervised learning that will give us the best match between these trajectories. This is a very tough process because, most importantly, there's not much data collected by the community that can support this line of work. Okay, so this is the motivation, and I'm hoping that in the next few years people will move in this direction, collecting data that then can be used for comparing neural networks and animals during development, right? And of course, the other complexity is that methodologically we are not there yet. We are very good in comparing snapshots of brain and neural networks, but when you have trajectories, you know, even mathematically, technically, it's harder task to do. So some methodological development needs to happen there. And yes, so thank you very much for listening. I also thank my collaborators, and yeah, I'm happy to answer your questions.
Thanks for your talk. And Any questions from the audience? Yep, sure.
Hi, yeah, thanks for the great talk.
Yes.
Yeah, sorry. So I was curious about your neuroanatomical determinism piece. So the way that you've implemented it now is to do some type of pre-training, right? So have you considered trying to look at like circuit motifs that you can only find in one or the other? And so then having like actual structural rather than like a kind of like structure functional type combination.
I love this question. So this is basically what we currently are doing. So pre-training is the right word, but with the current definition of pre-training, it's not exactly what we're doing. So we are pre-training for a very short period of time, which kind of nudge the network towards something. So if you look at, for example, filters, they're not yet formed, right? So it seems that the distribution of parameters in the network are shifting toward distributions that are good for a specific specialization. Now, one of the challenges that we have now is that what should we exactly look for, right? It could be that, as you said, like circuit motifs. But because still in the parameters of the network, there's a lot of noise. What you will see is a lot of noise, right? And the network is huge, so you see like a noise and a noise, and you need to extract some sort of circuit motifs from this noise. We're working on that, but this is the exact right question for the next step, because if you're talking about genetically encoded some asymmetries in the network, it should be a structural. So one thing I can tell you is that, for example, the first layer filters of the network is where I started from. Okay, maybe there is some asymmetry in the first layer filters. That would be retina in our case, that causes this this asymmetry. That was not really the case, but we're exploring that. Thank you.
What about, sorry, just as a quick follow-up. So what about something like circuit motifs that can create like a high pass, low pass filter, which you might expect to be different, like a right heart style?
Yes. So this is the step that I did before pre-training. So when we had this, large number of instances of networks that had this kind of like, asymmetry in dorsal ventral property, we said, okay, let's look at the kernel properties, the low pass, high pass filtering. And we actually saw the asymmetry in low pass, high pass filtering almost after initialization, right? But when I tried to manually handcraft these asymmetries in the network, it didn't work. Because, to just give you an inspiration to kind of intuition of why this didn't work, when you create these asymmetries in low pass, high pass filtering, you also have to make sure that the downstream layer is reading out from this asymmetry. If you only create that in the first layer, and then the next layer is not really reading out from that asymmetry, properly, throughout training, it will just be overwritten by backprop. And backprop, the one other problem is that backprop is just too strong. Whatever asymmetry you create, it will just overwrite it. So, but yeah, so.
Cool, thank you.
Problem. I hear you, but yes.
Okay, so thanks for your talk. My question is regarding the choice of self-supervised learning as a model. I understand that for some, it's closer to some of what we see in the nature, but let's take the example of human. Kids are not growing in the tagless environment. Some kids, well, the parents are really present and they're gonna interact with the kids, so there are gonna be some tags over there. And other kids, well, they live in the less welcoming environment. So how do you take into account those variations and the presence or lack of those tags?
Very good point. So yeah, maybe I described the two extremes. I said, okay, we have label for everything. We have no label at all. But as you said, the reality is somewhere in the middle. Of course, kids get some sort of feedback from their parents, from the environment. But at the same time, if you just look at like the amount of data that kids are collecting throughout the first 3 years of their development, it is it is not comparable with any sort of label data that you could put together, right? So it is very possible that kids, through self-supervised learning, they are learning some general representation of shapes, forms, motion, how their movement affects visual experience, right? They might learn this kind of general stuff, and then they would fine-tune it or maybe change it a bit with the feedback from the environment, from their parents and so on. So this would get us to what's called semi-supervised learning, right? So one problem with semi-supervised learning technically is that you will have two sorts of objectives. One is supervised, one is self-supervised, right? And you need to find a very efficient way of combining them together to make sure that they don't collapse. Because it's very likely that, this is the main challenge that semi-supervised learning has. It can happen very well that the model doesn't learn anything, because whatever the network is learning through self-supervised learning and supervised learning are just contradicting each other. So there are challenges, but I think you're right. Like this is more accurate in terms of how we need to develop models of development.
Thanks.
No problem.
Hey.
Hey, Patrick.
Great talk. I thought it was really interesting in your dorsal versus ventral plot, your initialization that, once you train, it seems like you have like a unimodal distribution of biases. And so that's surprising, because I would expect that, if, as we claim in the 2021 paper, you have like these two like very separate things which occur, you would expect like some bimodal distribution. Like one of them is always dorsal, one of them is always ventral, but sometimes the labels are mixed up. So can you explain that? And do you think that you need to add in something that kind of decorrelates the two streams somehow?
Yes.
To make it work?
So 2 points. What we showed previously, and what you referred to as the bimodal distribution of specializations, was after 100, 150 epochs of trials, epochs of training. This was only after 10 epochs of training. So many of the properties of the two pathways after 10 epochs of training are not yet fully formed. Some of the initializations, they are quite fast in acquiring the specialization that will, determine their final role. Here, because we wanted to generate all these samples of initialization, we had to limit the number of epochs of training. So because of that, yes, it's more unimodal. But one thing that we observed, and I didn't show here, is that a network that is dorsal-like after 10 epochs, that pathway stays dorsal-like after 100 epochs of training. So this is kind of a proof of concept that what we are doing methodologically is good. But ideally, you wanted to actually go for the whole 150 epochs of training, redo it for 200 random seeds, and come back 1 year later, collect your data and plot them. But this didn't happen. So to answer the other question, yes. So if we could do something like what people do in Barlow-Things, right? Decorrelate the representations of the two pathways throughout terms. So use it as a regularization term. That might force a network even to focus more on different eigenmodes of the input data. I'm not promising that it would be necessarily dorsal versus ventral, but it might focus on different eigenmodes of the input distribution of the data, of the the autocorrelation of the data. But yeah.
But do you think that right now in your two different pathways, like that the eigenmodes are like, is your claim that the eigenmodes are dorsal versus ventral right now?
So I'm hoping for that, right?
Nice.
So one thing that, there is a line of work showing that networks that are trained with backprop and gradient descent, the weights of the network will try to mimic the covariance matrix of input-output. This is Andrew Sachs' work and the follow-up works, right? So people have shown that when your network has multiple pathways, actually each pathway will focus on one of the eigen modes of the inputs, of the covariance. And this is something that theoretically has been shown. Now, does it correspond to ventral dorsal specialization is something that we couldn't test because we couldn't measure the covariance matrix. And the network is nonlinear, so we have to linearize it and look at the eigenvectors and eigenmodes. But what I'm trying to, so this is one of the things that we'll do next, is that just do it in a very simple problem. In a moving MNIST, everything controllable, and see if the network actually, what the dorsal ventral nest that the model learns corresponds to the eigenmodes of the data. That would be, ideal. So yes, that is the idea.
Thank you for your great talk,
Thank you.
and thanks to the audience for the questions.